Good morning. Thank you so much for your kind introduction. Thank you for coming. He said, I'm a group leader from the Max Planck Tandem Group in Nano-Bioengineering in University of Antioquia. Over there, we have 3 tandem groups, one about biophysics, another one about reproductive biology of the mosquito, Aedes aegypti, that is the vector of tropical disease infections, and my group that is about nano-bioengineering. So today, I divide the presentation in 5 topics. I'm going to talk about nanobiohybrid platforms for biosensing. I have 3 examples of how we can develop biosensors based on immuno, geno, or sugar-based biosensors. An example how we amplify signals in biosensing, and a couple of examples when we develop nanocarriers for a specific drug delivery. So our aim in our group is to develop hybrid platforms based on nanoparticles, carbon nanotubes, and any other nanomaterial, and we conjugate with biomolecules in order to develop platforms with enhanced properties for the diagnosis of diseases. We have 3 different research lines, one devoted to develop nanoproofs, biosensing platforms and devices for disease diagnosis, another one to study the host pathogen interaction through glycobiology and microarrays, and the other one to study or to develop different nanocarriers for drug delivery. So we envision a new generation of point-of-care systems with enhanced properties for the diagnosis of diseases. This device needs to be fast, of low cost, and accurate if we want to go a step forward in the development of personalized medicine. In our first research line, as I said, we conjugate different, sorry. Maybe it's the battery. So in these research lines, we conjugate different nanoparticles, nanotubes, nanofibers with bioreceptors, even proteins, antibodies, peptides, RNA, and so on, in order to develop enhanced materials or biohybrids. As a first example, I bring you this hybrid that is based on carbon nanotubes, decorated with iron and covered with some graphene layers. So in our first work, we develop this nanohybrid and characterized from the material point of view. So in this TEM image, you have the carbon nanotubes here, the iron nanoparticles, and the graphene covering the iron nanoparticles. We also perform differential thermogravimetric analysis to study the composition of the hybrid, and also Raman spectroscopy to check the graphitization process is not changing as we were assembling the hybrid, and also magnetization loops in order to demonstrate our hybrid has magnetic properties. And in the second work, we characterize the hybrid from the electrochemical point of view. So in this graph, you see here cyclic voltammetry. By cyclic voltammetry, we can also know, for example, how much hybrid can we deposit on top of a screen printing carbon electrode, and also we can follow all and characterize all the electrochemical parameters as we were decorating the screen printing carbon electrode with the hybrid step by step. We compare these cyclic voltammetry experiments with electrochemical impedance spectroscopy all the time. And the message here is with very small volume of samples and very short time, we can have a lot of analytical information. For example, we can have the peak intensity that we can relate with the electrochemical area of the hybrid. We also have different, this is the PDF? This is the PDF? Okay, so we are going to have problems. So in here we have, we also can have, for example, if it's a diffusion control process, or even we can have kinetics parameters with very simple experiments by electrochemistry. This is an example of a sensor for hydrogen peroxide detection, and the cyclic voltammetry, you see here how the current is increasing as the hybrid with form. And in this chronoamperometry, you can see better how the current intensity is higher as we compare with the carbon nanotubes decorated with iron and covered with graphene. It means the graphene layer is decreasing a little bit the current intensity, but is helping us to have a stable hybrid for even 1 month. This is a quickly example when we study, we develop an immunosensor. This is in collaboration with National University of Colombia. This is a veterinary pathology group in here that was worried about Streptococcus agalactiae. This is a bacteria that is killing a huge population of tilapia fish in fisheries. Along with them, we developed this immunosensor based on a single antibody. It's a biotinylated antibody that is we use for dual purpose. The antibodies recognizes the antigen that is expressed in the cell membrane of the bacteria, but also we're using this biotinylated antibody to anchor the bioconjugate in the surface of the screen printing carbon electrode, which is covered with neutravidine. So we use the interaction neutravidine, streptavidine, neutravidine biotin. So in here you have a SEA image when you can see all the bacteria confined in the electrode. After this, we may react with a streptavidin HRP complex, and we can produce an electrochemical signal in the presence of hydrogen peroxide and a mediator, and this electrochemical signal we can relate with the concentration of bacteria in a sample. So in here, this is a chronoamperogram when you can see the signal for a buffer that contains the bacteria with respect to a buffer that doesn't contain the bacteria, so we can distinguish contaminated and non-contaminated samples. So we need to characterize the immunosensor in all the steps of the fabrication process. First of all, we need to optimize the concentration of antibody, the incubation time, the concentration of streptavidin HRP, and incubation time. Once we optimize all the variables involved in the immunosensor, we can challenge the immunosensor with different concentration of bacteria. As you can see in this graph, we can build calibration curves with limit of detection of 10 cells. This calibration curve is from 10 to 1 to 10 to 7 column form units of the bacteria. And what is most important, we check selectivity. This is the signal of the immunosensor for Streptococcus agalactiae, and these little signals is when the immunosensor is challenged with another bacteria that can coexist with the Streptococcus in a sample. We went a step forward and challenged the genosensor in different samples. This is an environmental samples, spike with different concentration of bacteria, and this is indeed lake samples from a fishery here in Colombia, and we have statistically significant differences indicated that probably these lake samples were contaminated with the bacteria. This example is a genosensor. This is in collaboration with PESET in University of Antioquia. Lo siento. Sí, yo sé. I'm sorry. So this is a genosensor for detection of Zika. and we can differentiate from dengue and chikungunya. The diagnosis of Zika is very important, especially in pregnant women in the first 3 months of pregnancy, because the newborn can be delivered with microcephaly or another congenital problems. So the symptoms from Zika, dengue, and chikungunya are very similar, so it's very important to distinguish Zika from the other viruses. So for this purpose, we use magnetic nanoparticles decorated with neutravidine and attached to a biotinylated captor probe. This captor probe is specific for the target, for the RNA from the Zika virus, and is in sandwiching between a signal probe that is labeled with digoxygenin, and through the interaction with an antibody, anti-digoxygenin, labeled with HRP, we can produce the electrochemical signal, and we can correlate the concentration of the target present in a sample. So first that all, we need to develop the target sequence, the capture and signal probe sequences. For this purpose, we did a bioinformatics design and select, for example, the conserved region. From the conserved region, we select the target, the primers to amplify the target, and also the capture, biotinylated capture probe, and a digoxygenin labeled signal probe. After we design the primers and the capture and signal probes, we need to double check that the primers amplify the target. So we did this by PCR and qPCR. As you can see here, the signals are corresponding to amplification of Zika virus, and with the negative controls, no amplification for dengue and chikungunya. Once we double check the primers are working, we move to the assemble of the genosensors in a colorimetric assay. So we need to optimize again neutravidine concentration, capture probe, signal probe, concentration, and anti-digoxygenin HRP. Once we optimize all the variables, we can build calibration curves in the colorimetric format with a limit of detection of 9 picomolar. But also when we move to the electrochemical format, we can have similar detection limits. We also challenge the genosensor in close to real samples. In this case, we use saliva, serum, and urine, and spike with different concentration of Zika virus, dengue and chikungunya, even with a huge excess, 1,000-fold concentration of chikungunya and dengue, and also when the DNA strands were coexisting with other species from dengue and chikungunya, and in all the case, we only have signal for Zika. It means our genosensor is specific. We move a little bit closer to a real scenario. In this group, you have a statistically significant difference with synthetic DNA in the order of picomolar respect to a negative control. We also have signals for cDNA from patients and also RNA directly from patients. So this biosensor format is really promising. However, if you see here, the label of concentration is in the order of picomolar, and when we have an infected patient, the viral loading is in the order of femtomolar, so we need to keep working in improving the sensitivity of the genosensor and also lowering the detection limits. So for this purpose, in the same project, we developed a nano-bioconjugate. It's based on gold nanoparticles decorated with the signal probe and also a spacer probe that has biotin. So we will have multiple binding points through biotin to bind with this streptavidin HRP complex, so we will dramatically amplify the signal in this way. So we try with the genosensor in the nanoparticle format, but also assemble the nanoparticles on top of a screen printing carbon electrode, but by using ruthenium salt as an intercalator in between the DNA strands by electrostatic interaction, and uses this salt as an electrochemical probe. So in this way, we don't need to use the enzyme to amplify, but we use the ruthenium salt. So first of all, we need to optimize the nanobioconjugate and demonstrate that it's stable in physiological conditions. conditions. So for example, this is the plasmon resonance for bare gold and is shifting to the right when we code with the streptavidin and with double-strand DNA. We also study the effect of salt concentration, pH, hybridization temperature, and also the length of the DNA strands on the stability of the nanobioconjugate. We also study streptavidin HRP concentration, concentration of gold nanoparticles, signal probe, the ratio between the spacer and the signal probe, and also the hybridization temperature. And in order to increase the sensitivity of the genosensor, what we use is I can see here, but it's a screen printing carbon electrode decorate. A screen printing carbon electrode decorated with hierarchical gold nanostructures, and we assemble all the genosensor on top. So for this purpose, we electrodeposit a semiconducting polymer on top and electroplate these hierarchical structures. That is not here because it's not animated, the PPT. So at the end, what we did is by differential pole voltammetry, we can get a calibration curve with 0.04 femtomolar concentration. It means we are at the point that is relevant from the clinical point of view, and we are ready to test real samples. This is the last example of biosensors. In this case, we're using a glycophosphatidylinosytol glycan, I will call GPI. This is in collaboration with the Max Planck Institute of Colloids and Interface in Potsdam, specifically with Professor Daniel Baron, who is the next speaker. So they developed this GPI, and it has a tile group in one of the ends, so we take advantage of this tile group to chemisorb on top of a screen printing gold electrode. This GPI is able to recognize toxoplasma antibodies. Toxoplasmosis is a fungal infection transmitted by cats. So in the first step, we characterize the development of the sensing platform by electrochemical impedance spectroscopy. As you can see here, we have a little impedance related with the electron transfer of the gold electrode. This impedance is increasing when we functionalize with the GPI, and it's increasing even more when it's making react with a serum that contains the toxoplasma antibodies. This is the signal respect to a non-reactive serum. In here, we also optimize the concentration of GPI based on the signal to noise radio. And what is more important, we check specificity against another parasites like cruzi and falciparum is absolutely specific. We build calibration curves by impedance with a limit of detection that is 0.31 international units per milliliter, which is of relevance from the clinical point of view. When we compare our results with the gold standard ELISA assays for analyzing the antibodies, we demonstrate that we have only 5% maximum error with respect with this gold standard method. In the last research line, we have developed different nanoparticles and microparticles, even photosensitive to UV light or near infrared. They can be static or even self-propelled in a medium for different purposes, for biomedical or environmental remediation purposes. This is one of the examples when we encapsulate dofetilide. Dofetilide is a drug that is used to fight arterial fibrosis. So we develop a nanoparticle based on chitosan. Chitosan was modified with azobenzene, which is a UV photosensitive molecule. So we developed photosensitive nanoparticles, encapsulate the dofetilide, and functionalize the surface of the nanoparticles with a cardiac targeting peptide. This peptide is helping us to internalize the nanoparticles in cardiomyocytes in a very efficient manner. After internalization by exposure to UV light, the drug principle is going to be released inside the cells. So in the first step, we characterize the polymer by infrared solubility testing and also changing the concentration on monomer and also the water organic aqueous organic phase ratio in order to have different nanoparticles with different size. So we got nanoparticles from 90 to 600 nanometers. We characterize the size, the charge of the particles with the SEM and TEM. In this TEM, you can see here the peptide on top of the particles. We perform isomerization experiments, and we get almost 100% of isomerization after only 14 seconds, so it's very quick. We also try the specificity of the nanoparticles. The nanoparticles are functionalized with the cardiac targeting peptide, so we demonstrate that we can get inside the cell in even 20% after 8 hours interaction with the cardiomyocytes, as we compare with incubation even during 24 hours with the nanoparticles that doesn't have the peptide. So the peptide is very specific. We also try with different cell lines, and we demonstrate our targeting peptide is only internalized by the cardiomyocytes. We also perform drug release experiments. This is inside the cells. For this purpose, we co-encapsulate a dye, the dye and the drug. So after UV radiation, you can see here in red, the Nile red inside the cells as an indication the particles was destroyed, and the inside the drug and the dye inside the particles were released in the cytoplasms. We also studied drug concentration, dye concentration release, and also cell viability studies to UV light and to the concentration of nanoparticles that we use in the study. So to finish quickly, this is the last example. We're using polymeric biocompatible nanoparticles from PLGA and PLA to encapsulate drugs to fight intracellular infections. For this purpose, we are using Histoplasma capsulatum as a model of the infection. We're using itraconazole as a drug model. And at this point, we optimize the formulation. We estimate the drug loading capacity and the encapsulation efficiency. And we expect to functionalize with nanobodies as a part of the project. And we are establishing a collaboration with the Max Plan for Polymer Research in Mainz in order to perform a more basic study how interact the nanoparticles with the nanobodies with fluids. So once we have this study, we expect to have in vivo experiments here in Colombia in collaboration with the Biological Investigation Corporation, Corporación de Investigaciones Biológicas, to study pharmacokinetics, biodistribution, and histopathology. And finally, I don't know if I have time for concluding remarks or I go directly to the acknowledgements. I want to acknowledge the organizers of this symposium. I would like to ask to thank Colciencias, University of Antioquia, the Max Planck Society for the support, Ruta N for hosting our tandem groups in Medellin, and also thank you for your attention.